trying to recover from eating too much. It's front and back, so make sure you do both sides. And one is a genetics problem, the same kind of thing that you'll see on exams, and then the other side is some questions relating to meiosis and mitosis. Okay, so make sure you do that. Also, an announcement, the study session tonight will be the last study session. Your student instructor is being shipped off to Kuwait, so she will no longer be here to do study sessions. So tonight will be the last one. However, what I'll try to do is set up during Dead Week. I'll try to set up a study session or two that you can come to and ask any questions if you need to. Okay. The first thing I want to do today is clear up a little confusion about what chromosomes do in meiosis and mitosis. Last time we looked in some detail at mitosis and meiosis and how they work and what chromosomes do in order to separate genetic material. So what I just want to do is kind of show you how you can count chromosomes and chromatids when you're going through all these stages of meiosis and mitosis. So if we start out in G1 phase, if you could see the chromosomes, and you can't because they're too diffuse, but if you could, they're all scrunched down and compacted, this might be what we would see. This is a situation where 2n equals 4 or n equals 2. Remember that 2n equals 4, that's the diploid number of chromosomes. That's when you have 2 copies of everything, as this organism does. N refers to each set, so n is half the number of the diploid set. Okay, so we're saying a set of chromosomes is either the 2 blue ones or the 2 black ones. All right, now when you go through S phase, at the end of that DNA synthesis phase, your chromosomes would be duplicated. Okay. So we would see something like this. All right. We haven't doubled the number of chromosomes, though. If we count chromosomes in this first G1 picture, we've got 4 centromeres. So you just count centromeres to count the number of chromosomes. All right, so the number of chromosomes equals the number of centromeres. So in this case, we've got 4 centromeres, so we've got 4 chromosomes. And while it's not really appropriate to refer to each of these as a chromatid, we sort of can for conceptual purposes. So we can say that there's 4 chromatids. In this next picture where we've gone through S phase, we haven't changed the number of chromosomes. Okay, so the number of chromosomes, if you count centromeres, it still equals 4. The number of chromatids, however, is what's doubled. We now have 8. Right? 1, 2, 3, 4, 5, 6, 7, 8. Because all these strands have doubled. All right, so these go through prophase and metaphase. If we're looking at mitosis, for example, they go through prophase, they go through metaphase, they line up on the metaphase plate, and then, so let's just draw that. We've got our chromosomes lined up on the metaphase plate. Okay, we still haven't changed the number of chromosomes. But in anaphase, what we're going to do is we're going to separate centromeres so that chromatids go to opposite poles. Okay, and just pull those strands apart. The result of that is that we can end up sort of conceptualizing this as being, as soon as you get into telophase, we have 2 separate cells where we've gotten these chromosomes separated. Okay? So we go through telophase. These cells are going to divide by cytokinesis, and you end up having two daughter cells that clearly have the same amount of genetic material as the original parent cell. Okay? So each one has two blue chromosomes, and each one has two black chromosomes. You haven't changed anything. Right? So really it's anaphase where you separate out the numbers so that they become the same as they were before. So that in telophase we end up again having 4 chromosomes and 4 chromatids. Okay, so 4 chromosomes, 4 chromatids. Situation is a little bit different in meiosis, though. If we go through meiosis, chromosomes pair. Right? That's the major difference. Okay, so for meiosis, if we start out with our the same chromosomes that we had and line them up on the metaphase plate, what we end up happening is having pairs. Okay, so there's our pairs of chromosomes. All right, so we still have 4 chromosomes, 8 chromatids, and remember these pairs of chromosomes are called tetrads or bivalents. We have 2 tetrads. And remember the term tetrad and bivalent are interchangeable. So 2 tetrads, 2 pairs. When we separate those in anaphase, we don't separate centromeres, we separate pairs. What does it say under here? Bivalent. So in anaphase, you separate the pairs. We end up getting 2 cells. In this case, the arrangement that I've drawn means that the black chromosomes went to one pole, and the blue chromosomes went to the other pole. Okay? So if we visualize this in telophase, as the cell division, cytokinesis is occurring, we end up getting 2 cells, one of which has the black chromosomes, and the other one has the blue chromosomes. All right? Now, important point here is if we're counting chromosomes and chromatids, we've halved the number of chromosomes. Right? In each of these cells we have 2 chromosomes. We have 4 chromatids. Okay? 2 chromosomes, 4 chromatids. That's why that first division is called the reductional division. You reduce the number of chromosomes. You half them. Okay? Beyond this, the next division is just the same as mitosis. So if you go through all of those stages, prophase 2, metaphase 2, anaphase 2, and telophase 2, okay, you don't really change anything except the number of chromatids. The number of chromosomes stays the same. Right? Those cells are going to retain 2 chromosomes. It's just that in anaphase you're going to separate the centromeres, just like you did in mitosis. So you end up having 2 chromatids instead of 4 chromatids. Right? So 2 chromatids, 2 chromosomes per cell. Right? Sometimes it's easier to see this just by drawing it out. Okay, so that's how you count chromosomes and chromatids when you're going through meiosis, and there was some confusion on that, so I want to try and clear that up. Does that make sense? All right. I'll assume by those vacant stares that it does. Or it's so unclear you don't know what to ask. It could be a possibility. Okay. Okay. So here, as I showed towards the end of the last period, are the major differences between mitosis and meiosis. All right, in mitosis, we start out with prophase. We've got duplicated chromosomes with sister chromatids. Okay, so right up here at the top. There is no pairing in mitosis. So these chromosomes independently line up on the metaphase plate in no particular order. In anaphase, we separate centromeres. So chromatids, these chromatids are going to go to opposite poles. They're going to be pushed and pulled apart. All right, the result is that you get daughter cells that have the same number of chromosomes as the parent cell. Okay, however, in meiosis, the fundamental difference is that chromosomes pair to form tetrads. And that's what this term synapsis refers to, as formation of pairs. Okay. All right, so we have pairing in meiosis 1. And in meiosis, at the end of meiosis 1, we separate pairs instead of centromeres. That's what halves the chromosome number, as is shown here. So now these daughter cells have half the number of chromosomes, and all we've got to do is separate those chromatids so that everybody has the same number of chromatids. And that's what happens in the second division of meiosis. It looks just like mitosis. These chromosomes line up on the metaphase plate. You split the centromeres, and the strands go to opposite poles. So you end up with 4 daughter cells, each with half the number of chromosomes of the parent cell. All right, the critical difference there is the pairing. All right, if you remember that they pair in meiosis 1, then you'll never go wrong, because you have to have pairs, and those pairs then have to separate, and then you can separate chromatids. Right. Okay, so as I said, when chromosome behavior was observed, it appeared to be very similar to the behavior of Mendel's unit factors, genes. Okay? People made the connection very quickly, and they supposed that genes were in fact on chromosomes. It turns out they are, because chromosomes are long linear strands of DNA, and DNA is the genetic material. Okay, so can the behavior of chromosomes account for segregation? The answer is yes. Are genes on chromosomes? Yes. We can demonstrate this sort of intuitively, but there are ways of actually demonstrating that it's true experimentally. That's really beyond the scope of this class, but we can do it. When chromosomes pair and they line up on the metaphase plate, there are alternate arrangements that could occur. In this organism, there's 2 different arrangements. Either the blue chromosomes are on one side and the red are on the other, or you have an alternation, as in possibility 2. Okay, so when you separate those tetrads in meiosis 1, in the possibility 1, we end up with all the blue chromosomes in one cell and all the red chromosomes in the other cell. Then in meiosis 2, you simply separate out those centromeres, okay, just like mitosis. So here's two combinations that can result from the first possibility. All the blue chromosomes in a couple of cells, all of the red chromosomes in a couple of other cells. The second possibility, though, has a blue chromosome and a red chromosome in each. And so you end up with the other two possible arrangements here at the bottom that you would expect to see if you were looking at segregation of Mendel's unit factors. Okay, so we can actually put genes on these chromosomes and ask, does it work? And this is actually what I showed at the end of the class last time. Let's suppose that we're looking at an individual that's heterozygous for round and yellow seed traits. So it's big R, little y, little y. Remember that genes are in the same place on homologs, but you don't necessarily have identical alleles. So that's why the R's are in the same place on those big chromosomes, and the Y's are on the same place on little chromosomes. Two different possibilities. We could either have big R and big Y on one side of the metaphase plate and little r and little y on the other, or we could have an alternation. Okay, over here on the right, little r, big Y, big R, little y. When you separate those pairs, they're going to separate such that in this left-hand possibility, big R and big Y end up in one cell, and little r, little y end up in the other cell. Okay, the other possibility here on the right, you get little r, big Y, and big R, little y. So if you follow those through the second division of metaphase, or sorry, the second division of meiosis, you end up getting equal probabilities of each of these four gamete types, okay, equal frequencies. So we get 1/4 big R, big Y, 1/4 little r, little y, et cetera, et cetera. That's exactly what Mendel observed with unit factors, the segregation of unit factors. They're in pairs. They're separating out so that each gamete gets one or the other, but not both. Okay? And so as a result, you have equally likely combinations. When you get fertilization in the F1 plants, then that's where we saw our 9:3 to 3:1 ratio of segregation. Okay, so it was clear that Mendel's unit factors did behave exactly the way chromosomes did. It remained for another 10, 15 years before it was distinctively proven that genes were in fact on chromosomes. Okay. It is also known that genes are arranged in a linear fashion on chromosomes. Okay. Think about this. Nobody knew what chromosomes were like. I mean, I've told you basically the answer before all of the data to support the answer. We know that DNA is a long linear strand. Okay. That's what chromosomes are made of. Each chromosome is a single linear strand of DNA. We know that a gene is a sequence of bases in DNA that specifies a particular protein. And we know that now, but they didn't know that then. They had no clue about chromosome structure. They could have been these big three-dimensional tubes where you had genes just scattered throughout those tubes. We know that that's not the case now. They are arranged in a linear manner. Okay, we can, in fact, map them. Right? There's this funny observation of crossing over, that chromatids, non-sister chromatids, will actually pair and exchange pieces of themselves physically. When they do that, alleles that are dissimilar on those chromatids will cross over to the other chromatid. That's why it's called crossing over. Okay? Crossing over is a statistical phenomenon. So the closer two genes are on a chromosome, the less likely they are to cross over. So you can actually measure how far apart they are by measuring how much they cross over. Again, that's going to be beyond the scope of this class, but that's how it was demonstrated that chromosomes were linear, that genes were arranged linearly on chromosomes, by observing crossover frequencies. Okay, crossover is an important tool as well because it allows us to map markers to genes that cause, say, human diseases. human genetic diseases, so we can isolate the gene that's causing that particular disease. Okay, non-disjunction. Normally, chromosomes separate so that the pairs separate normally, and then the chromatids separate normally in the second meiotic division. That process is called disjunction, right? The separation of chromosomes normally during cell division is disjunction. Okay, and oftentimes we'll actually say chromosomes disjoin. Non-disjunction then is the abnormal separation of chromosomes, a failure typically of chromosomes to separate. Either the pairs might not separate in the first meiotic division, or chromatids may not separate correctly. Okay, either one is possible. Okay, so it can occur in meiosis 1 or 2. It can also occur in mitosis. Okay. As I say here, mitotic non-disjunction is usually not a problem. You have a population of trillions of cells in your body. If one of them happens to divide and you get non-disjunction occurring, it's not usually going to be a problem. In fact, that cell will probably die. Right? If it occurs very early in development, at say the 2-cell stage or the 4-cell stage, then it can be a problem. Having an excess number of chromosomes in half of your cells, or not enough chromosomes in half of your cells, can be a significant issue. All right, so here's an example of non-disjunction. This non-disjunction is occurring in meiosis 1. So what we're looking at up here at the top is the second pair of chromosomes, which is failing to separate in meiosis 1. Okay, that produces 2 cells that are lacking or have excess number of chromosomes. Okay, so this cell on the left has too many copies of the large chromosome. It has 2 when it should only have 1. And the cell on the right is lacking a copy of the large chromosome. Okay, when you go through meiosis 2, it's very rare for non-disjunction to happen twice in the same meiotic event. Okay, so if meiosis 1 suffers a non-disjunction event, it's typical that meiosis 2 would be normal. So we separate these chromosomes normally, and we end up having 2 gametes that have an abnormally high number of the large chromosomes, and then 2 cells that are lacking the large chromosome completely. Right? So if we are looking at number of chromosomes, N, that's the haploid number, plus an extra chromosome. So that's where that N plus 1 comes from. Here, N minus 1 is the haploid number of chromosomes, 1 copy of each, minus the 1 that didn't get in there. Okay, so these can cause some serious problems. If the event occurs in meiosis 2, however, then only half of the products are abnormal, but it's still a big issue. Suppose that meiosis 1 occurs normally, so we disjoin these chromosomes correctly, the pairs, but then in meiosis 2, we fail to separate these chromatids. As a result, you have a gamete that, again, is N plus 1. It's got an excess large chromosome. A gamete that is N minus 1 is lacking large chromosome, and then the other 2 are normal. All right? So you can end up having all of the products of a meiosis abnormal, or only half the products of a meiosis abnormal. Okay? This phenomenon of non-disjunction leads to some serious consequences. Down syndrome is a result of a non-disjunction of chromosome 21 in humans. Okay? Down syndrome, you would think, is relatively less damaging than some others. Individuals live to be about 40 years old, for example. They typically have internal malformations, varying levels of mental setback as well. But there are more serious things that can happen. A non-disjunction of chromosome 13 or chromosome 18 will lead to either Patau or Edwards syndrome. Those kids don't usually live more than 6 months. Right? Non-disjunction of chromosome 9, if you have 3 copies of chromosome 9, you're lucky if you make it to birth, and you're not going to live for many more hours after that. It's really serious. There are serious internal malformations that occur. So those individuals don't live very long. All right, so these non-disjunction events have serious consequences in terms of human health.
So why does the Down syndrome, why does it show up so often? That particular one?
Good question. Why does Down syndrome show up so often? It's because it's the most survivable of all of the non-disjunction events. Right? The reason, if you look at spontaneous abortuses, you take a sample of 1 million spontaneous abortuses and ask, what are the chromosomal abnormalities that you see? You see everything, except for non-disjunction events that involve the biggest chromosomes, like chromosome 1, 2, 3. Those chromosomes just have too many genes on them. And as a result, that individual usually doesn't get past, say, the 16, 32 cell stage. They don't even implant, typically. And when they do implant, right, and you get a spontaneous abortion, the abortus didn't make it very far. It's a very early embryo. Okay? So that's the reason why you see them so often. They're just the most survivable. There are very few situations in humans where you can survive only having one copy of a chromosome. A lot of times 3 is better than 1 in terms of survival. There is a syndrome called Klinefelter Syndrome in humans that results from a partial lack of a particular chromosome. I think it's chromosome 5. So you're missing part of that chromosome. It's called Klinefelter because the individuals that have it have a defect of the larynx that when they cry, it sounds just like a cat meowing. It's the most bizarre thing. But that's why it's called Klinefelter, right? Cat cry syndrome. Most what we call monosomies, situations where you only have one chromosome, are not very survivable in humans. Okay, we're going to move back then into chapter 9. Are there any lingering questions on meiosis and mitosis anybody wants to ask before we move into chapter 9 again? All right, I want to spend some time talking about sex determination. It looks like that next week I'll be able to expand some more on some of these genetic topics, which I don't usually get to do. It'll be kind of fun. Sex determination is a genetic process. What we're talking about is determining whether the individual is going to be male or female genetically. Okay? That's where the cells establish their sexual identity. This individual cells establish whether they're going to be male or female. And there are differences in gene expression between those two states. Okay? Differentiation is a much different process. That's where the genetic state is realized. That is, you have genes that are activated in a male pathway or a female pathway. And those active genes then determine whether that individual develops as a male or as a female. That's differentiation. There's 2 levels of that. Primary differentiation is developing all the plumbing. The second level is developing all of the characteristics that appear at puberty. That's at sexual maturation. Okay? So those are the 2 levels of sexual differentiation that occur. Environment can play a role as well. Right? Gender identity is a lot more complicated. By gender identity, I mean how do you identify yourself? Do you identify yourself as a male or a female or somewhere in between? Okay? Those sorts of issues, interestingly, are to a large extent programmed by brain development. There's different wiring in the brains of males versus females. There are physically different characteristics in the brains of males and females. We don't think the same way, which is really interesting. But that is largely influenced by genes, and that brain development process is all a part of the development of the fetus. Okay, so we might have a chance to be able to go back and look at some of the issues involved with the genetics of gender identity. It's really interesting. Okay, so how is sex determined? What do you have to be a male or a female? Well, the answer depends on what species you are. Okay, most of us know that in humans you have to have a Y chromosome if you're going to be male. If you lack a Y chromosome or a very specific piece of it, you are female. But in other species it's not so clear. So there's a lot of different modes or methods of determining the sex of an individual. Right? Up here at the top, in this grasshopper, we're showing a situation where you have either one X chromosome or you have two X chromosomes. If you have one X, you're male. If you have two, you're female. We call that an XXXO system. The O is not referring to any chromosome. It's just referring to the absence of that second X. Okay? So that's why it's called XO. There are other situations where it's exactly the opposite. An XX might be a male, and an XO might be a female. Again, it depends on the species. In birds, the male is ZZ. Okay, so it's got two Z chromosomes. The female has a Z chromosome and a W chromosome, kind of like the X and the Y in humans, except here it's opposite. All right? In humans, an X and a Y gives you a male. In birds, a Z and a W gives you a female. There's yet another system. This term heterogametic. Heterogametic refers to the types of gametes you produce. In the case of birds, because this bird is a ZW, it produces two different kinds of gametes. One type that has a Z in it, and the other type that has a W in it. The Z and the W pair in meiosis, and so they separate just like any other pair, and that means that gametes will either have a Z or a W. In humans, very similar, except the male is heterogametic. The males have an X and a Y, that X and Y pair, and so the gametes can either have an X chromosome or a Y chromosome. Right? So all male offspring get their Y from their fathers and their X's from their mothers. Females get an X from the father and an X from the mother. Okay, in Hymenoptera, that's the insect group that includes bees, ants, wasps, etc. It's the number of chromosomes that you have. If you have two copies of every chromosome, then you're a female. If you only have one copy of every chromosome, then you're a male. Okay, so in bees, the workers all have two copies of each chromosome, and the drones only have one. It's really interesting how this is achieved. The queen bee fertilizes most of her eggs, and in fact, she stores sperm for her entire life. Ant queens are the same way. An ant queen may live 30 years and use sperm for that entire 30 years from a single mating. And so if she fertilizes those eggs with a sperm, then you get females out. If she doesn't fertilize them, you get males out. There's lots of environmental cues and signals that can cause the female to stop fertilizing the eggs and cause them to develop into males if males are needed. Okay? If you're developing a new batch of queens to expand elsewhere, then you'd need some males as well. In ants, it always happens at certain times of the year. Typically in the spring here, you'll see ant nests where you've got lots of winged individuals. Those winged individuals are the reproductive ones. Okay, so there's environmental cues that cause the female to start producing unfertilized eggs, and that gives rise to the males. Okay. Life as a male hymenopteran is not good, though. The drones they just suck up resources. They don't really do anything except when they mate. Okay. In ants, a male will mate once and then he dies, and the reason is because when he disengages from the female, the reproductive organs just rip right out of the body cavity along with the male. There's a reason for that. What it does is it blocks the female's reproductive tract so nobody else can mate with her. I mean, that's a hell of a way to go. What about most mammals? All right, mammals typically are XXXY. So that's what we're showing in this figure. A male has 44 chromosomes plus the X and Y chromosome. A female has 44 chromosomes plus the two X chromosomes. Notice how sperm are being produced here. Those sperm either have an X or a Y. Okay? Females can only produce eggs with X's in them. So if you have an X-bearing sperm that fertilizes an egg, you get a female. And if you have a Y-bearing sperm that fertilizes an egg, you get a male. So as I said, the male offspring in mammals always receive their Y from their father and their X from their mother. Females get an X from each parent. Right? So this is what's called the XXY system, and it's true of most mammals. Right? The Y chromosome determines maleness. If you lack a Y, you are not a male. In fact, it's not the whole Y that you need. There's a little piece of the Y chromosome that determines maleness. That piece can actually get stuck onto an X chromosome, so you can get masculinization of a female, an XX female. It's really interesting stuff. All right, how do we know that the Y chromosome is male determining? Because we can look at sex chromosome abnormalities. Nondisjunction doesn't just affect all those other chromosomes. It can affect sex chromosomes too. Right? So you can get funny combinations of sex chromosomes. And it's those funny combinations that tell us something about how the sex chromosomes work. Okay? Most of these are not lethal. Okay? Sex chromosomes are less critical in terms of survival than all of those other chromosomes are. So if you have extra X's or Y's, it really doesn't matter. In fact, a fair number of females have extra X's and don't even know it. All right? You might experience menstrual irregularities, and that's about it. All right, so we can see some of these combinations. Triple X's, XXY, XO, XYY, those are all fairly common sex chromosome abnormalities. Okay. This one is typically referred to as a super female because it's got one extra X. These are referred to as super males because they have an extra Y. All right, so the sex chromosome abnormalities that we see are summarized in this table. The XXY is called Klinefelter Syndrome. It's named after the doctor that first described it. Right? It can arise from meiosis, failure of meiosis in eggs or sperm. That frequency is grossly incorrect. 1 in 2,000 is actually closer to 1 in 500 to 1 in 700. And it's much higher than this figure implies. XYY is also fairly common. I'd say that 1 in 2,000 is a little bit of an underestimate. Metafemales are common. That's about right, 1 in 1,000. And the Turner syndrome, much different. Look at that. 1 in 5,000 on average. Right? 1 female in 5,000 is a Turner female.
What happens if you sue?
We'll go through that. We'll look at some of the characteristics of each of these. They all have distinct characteristics. But yeah, it's a good question. What happens in all of these? All right, so XO is much rarer. Why might this be? The reason is XOs don't live very well. Right? They abort spontaneously more often than any other of these sex chromosome abnormalities. Okay? They have a tendency to take on fluid in the womb, and so as a result, they fill up so much with fluid that their cells don't function, their organs don't function, and they end up dying. They can't get rid of all that excess water. I've seen a lot of spontaneous abortuses of XOs that were just, they looked like fluid-filled bags with faces and limbs. It was pretty sad. Okay, so that's why XO is much rarer. All right, here's an individual with Klinefelter syndrome. As I said, it's much more common than your book implies. You see it 1 in 500 to 1 in 700 male births. It's the most common sex chromosome abnormality. Okay, so most common chromosomal abnormality of them all. All right, these individuals are clearly male. They have male genitalia. They have male plumbing. Right? So they clearly developed as a male. I refer to that plumbing as male and female, but it turns out that an embryo starts out with both. You've probably heard the phrase that embryos start out female. They don't. They start out neutral, or both. They have both initial sets of plumbing, or ductwork, and then you get rid of one set of ductwork and develop the other when the determination event is made to be male or female. All right, so these individuals are male. Clearly the external genitals are male. The testes are small, however. Usually they're undescended. Testes and ovaries develop in the same place in the lower abdominal cavity, and in the case of testes, because sperm development doesn't occur at normal body temperatures, it's too hot, they have to descend below or outside of the body cavity, so they descend into the scrotal sac. And often in Klinefelter's males, those testes simply fail to descend, and so they're sterile. They're typically sterile even if the testes have descended. They just don't develop correctly. Right? At puberty, they start developing the secondary sexual characteristics of females. They get breast development. They tend to have female patterns of fat deposition, which are different than males. They tend to have a female pubic hair pattern. So all those characteristics are female rather than male. So the determination event was male. But the other events, the differentiation events that occur, are clearly female instead. Okay. They're usually sterile. Okay. There are higher levels of this abnormality that exist, having 3 X's and a Y, 4 X's and a Y, 2 X's and 2 Y's. All of those you see in clinical genetic situations. They're much more unusual than the typical XXY. Okay. We do see this in other mammals as well. There are things like Klinefelter's dogs and cats and that sort of thing. All right, this is a female who has Turner syndrome, XO, much rarer than any of these other sex chromosome abnormalities because they don't survive to birth as often. It's a much harder condition to survive. And they tend to swell up with fluid, which is called edema. Right, that swelling with fluid. All right, they're female. Clearly they are female. They have a female external genitalia. The plumbing is female. Okay, they have underdeveloped ovaries, but everything else is okay. They do have some internal malformations. For example, this aortic constriction that you see. A characteristic sort of soft facial features. They typically have a web of skin around the neck, and there are elbow malformations which aren't being shown here. Right? Not every Turner female has all of these traits. They also tend to have varying levels of mental retardation. Puberty characteristics are poorly developed. They don't develop the pubic hair. They're going to be sterile because the ovaries have degenerated. Interestingly, in mice, it's not as severe, and they can actually reproduce for a short period of time. Right? There's a tendency towards mental retardation, but I've had Turner syndrome people in my classes that were just as sharp as anybody else. So a lot of variation there. All right? Okay, well, what about XXX and XXY? They're really not much of a problem. A super female is completely normal. They may have menstrual irregularities, but they do display a higher frequency of sex chromosome abnormalities in their kids. Because you've got 3 X's, and the 3 X's can't separate so that everybody has the same number of X's. So you end up getting gametes with 2 X's in them. XYYs are also completely normal. They tend to be a little taller than usual. They used to be thought to be more aggressive than normal. That turns out to not be the case. That's not true. Okay? All right, so what does this tell us? If you have a Y chromosome or a critical piece of the Y, you are male. And if you don't have it, you're female. Never mind all the differentiation events that occur. If you have the X, you're going to have male genitals, you're going to have male plumbing. Your cells are male and not female. Okay, so we'll stop there and we'll look at the segregation of X and Y traits next time. Yes?